everyone. Thank you so much for joining us today. Before we officially kick this off, I do want to point out to the audience that we welcome questions. Please post them in the Q&A section at the bottom of your screen, and we will address those at the conclusion. So I'd now like to turn this over to Steven Saltzstein, Forrest Family Office's CEO.
Thank you, Callie, and thank you very much, everyone, for joining us today. We have an incredible symposium for you with a company called Novel Bio, that is revolutionizing the world, and an incredible CEO who is absolutely a visionary, Pankaj Khanna. So, Pankaj, just to begin with, tell us a little bit about yourself, your background, and then how you got the idea for the company.
Yeah, sure. So I'm Pankaj Khanna. I'm co-founder and CEO of Novel. I'm a chemical engineer by training, by trade, and about 2 decades of experience in the biotech space. I started my career with Pfizer, then worked for CSL and Sanofi, worked on all different domains of biotech, or verticals, if you call, of biotechs. Worked on vaccines, on monoclonal antibodies, cell and gene therapy, as well as on human blood plasma as well. And most of the experience has been focused on technology development and commercialization, scale up and commercialization, where we look at a technology developed in a lab, how we can scale it up efficiently, and then actually put it as a manufacturing setup to manufacture those drugs. So that's what my sweet spot is. Very well versed with CDMO operations, have built many CDMOs, and also with GMP manufacturing. So that's where I kind of excel. And before founding Novel is my third venture in this space. I founded 2 other ventures. Both are successful ventures, profit making. First one was founded almost a decade ago. We provide project management and construction management services to the pharma biotech industry. And we have large and small clients where we have built a lot of vaccine facilities and CDMO facilities and labs for biotech and pharma. And the other one is a consulting business where we provide commissioning and qualification and regulatory compliance services. Between the 2 businesses, we have more than 150 employees and offices in North America, as well as Asia and Australia. So Novel is my third venture in this space. And coming, yeah, go ahead, please.
Well, just tell us what Novel does and how you got the idea for it.
Yeah, that's a great question. So during the COVID pandemic, everybody was on the biotech side was pretty engaged. We were actually very busy. I was personally very busy in setting up, as part of Operation Warp Speed, and was setting up plasmid DNA manufacturing facilities as well as filling facilities for COVID vaccines. So there was a lot of demand for plasmid DNA at that time. And what I realized was that the lead time for the DNA was going into years, 2 years, 3 years, if you want to secure DNA. And plasmid DNA is a starting material for not only COVID vaccines, that was the high point at that time, but it was for all cell and gene therapy, for any RNA therapy beyond vaccines, for direct DNA therapies. Any modern life-saving medicines starts with plasmid DNA. So those lead times were causing lack of innovation or disrupting innovation in those spaces because everything was going towards COVID vaccines. And the response, industry's response was, let's build bigger facilities. And we were part of that. We were helping build those facilities. And what we realized was kind of a little bit shocking as well, that everybody was using across the globe the same technology, 50 years old technology, E. coli based. And the industry's response to these issues were that my facility is bigger than yours. So just building a bigger facility with the same technology. So that led to an idea that why don't we look a bit differently? Because based on COVID's demand and response, the whole cell and gene therapy space and mRNA space was under the spotlight. And it was, very, I would say, given that the plasmid DNA demand is going to continue to expand. So it was right time to kind of enter the market with a differentiated technology that can solve the pain points. And that led to starting of Novel. So Novel Bio is completely focused on DNA manufacturing, DNA cloning and plasmid DNA manufacturing. That is what we do overall.
And so, how do you do it? How is it different from the current standard?
Yeah, so I think I would take you back a little bit on like how the DNA market developed and why people have been using E. coli ever since and how we are different. So I think that's very important to know. So
Please.
E. coli has been used for the last 50 years to make plasmid DNA. And the reason is E. coli is the most studied organism. It is the bacteria that people get trained on when they do their PhDs and postdocs. A lot of publications are out there on how to grow it, how to mutate it, what it likes, what it doesn't like. People know everything about it because it has been studied so much. So when these therapies or this domain got started, people started using E. coli to make plasmid DNA or DNA, cloning DNA. And they never kind of looked elsewhere because it was a well-known platform and it worked decently. As the industry expanded, that more tools developed around this platform. Now, before the sequencing of DNA would take an equipment size of a room to now it's a benchtop equipment. So everything kind of developed gradually around this platform, but people stuck to E. coli. The DNA designs in the beginning were very simple, small DNA designs, because the industry was just evolving. Over the 5 decades, the design became really complex and issues started cropping up. And I would say the COVID time was kind of the watershed moment where everything kind of came together and put the spotlight on these new therapies and the power of these new therapies. And it became very clear that E. coli was the bottleneck, that it has high failure rates. The cost is very high to make DNA. It's the single largest cost component. And also it takes a lot of time to grow. The batch times are really, really long. So all of that led to those pain points. And when we entered the field, we had basically a couple of criteria on what to do here. We thought we need to kind of move away from E. coli because everybody is using the same technology. And we thought there are multiple bacteria that have been discovered for the last 50 years that can do a better job of making DNA. Our criteria was the bacteria should grow really quickly because then it will reduce the batch times. It should have some viability or demonstrated in the previous studies that it can make protein or DNA or something like that, so that we're not starting from zero. There is something out there. And the third piece was it should be safe. It should be BSL-1 so that we can use it as an industrial, kind of factory. If it is BSL-2 or higher, then it is a disease-causing bug and we cannot really use it. So those three factors, we looked kind of into the database and we found a bug called Vibrio nitrogen. So Vibrio nitrogens is a BSL-1 bacteria. It is the fastest growing bacteria on the planet. And that is what we use to build the technology that we have now, kind of platform that we have developed.
What percent of the life, and you may not know this, I'm throwing you a curveball here, but what percent of the life science market, uses this delivery mechanism or could use this delivery mechanism?
So the life sciences market right now, there are more than 2,000 clinical trials in the cell and gene therapy space alone. And I would say it's about 20 to 25% of the market right now, but that is where the growth is. So there have been three, I would say, waves or kind of, I would say, iterations of this, you can call it, where the first one was small molecule, where, you take the over-the-counter pills now. The second one was this large molecule drugs, million monoclonal antibodies was the second revolution of biotech.
Right.
And then and both of them have kind of reaching at their end point that whatever could be achieved through those technologies has been achieved, and what else next now, right? So the third revolution is cell and gene therapy and mRNA therapies, which is coming together now. And it's increasingly more and more therapies or the clinical trials are for these through these mechanisms. So it's going to keep on replacing these existing drugs, and most of the new or unmet or the diseases that have not been, they don't have the cure, will be cured through these new therapies, modern therapies, going forward. So this is the future, in simple words.
Why do you think it took 50 years for some genius to come along and say, this is the biggest bottleneck in life sciences, like, let me figure out a different delivery mechanism?
Yeah, that's a great question. I pondered over it myself too. There are a combination of factors. There are actually, if you think about it, are two main factors. One is that prior to COVID, how many of us had heard of mRNA, right? And so it was not in the limelight, and the existing technologies were enough. Just like Tesla came up with an electric car. Electric cars were there.
That's a great analogy.
Yeah, the concept was there before too, but something led to, okay, we're going to change something. It was the oil prices kind of went through the roof, and it was like, why don't you look at something else? Or the global warming, both of factors combined together led to the electric car. The same thing is with this, where people were using E. coli, they were frustrated, but it didn't enter the mainstream. And the reason was that the focus wasn't there. And COVID right off changed the perspective and said, okay, this is the future. It kind of put a stamp on it, because before, mRNA technology is not new. It existed a decade ago. But the COVID pandemic validated that technology. And when it got validation, everybody started moving towards it and the market started expanding. And now we are not the only ones who are trying to develop plasmid DNA manufacturing. There are others who are trying to develop it through enzymatic process, not through another bacteria. But
Right.
they've entered in this market, but they entered mostly post-COVID. It kind of gained the limelight. The second aspect is why nobody tried to do it is very hard to do it, because everybody knows how E. coli functions and they get trained on it during their postdoc, during their PhD days,
Right.
and so many publications. So it's like a comfort zone. To use a new non-model organism requires a lot of work upfront, which many people are not willing to put. Those are the two main reasons.
And what DNA designs were previously unmakeable with E. coli that now you guys can?
Yeah, so that is a complex question. So on general level, the designs that have a lot of repeat sequences, we call it inverted terminal repeats, ITRs, are typically hard to do in E. coli. So what happens is E. coli typically doesn't like it, and it will either truncate those designs or it will change it to survive. And then some designs are inherently toxic to E. coli. Those are the two that have most of the problems in
Right.
terms of E. coli would not make it at all. Then there are others where the yields are low. And because of that, the cost is very high. Because the yields are low, you have to ferment a big batch and then to process a big batch, then leads to cost increases. So the issue is not only of batch failure that E. coli just cannot make it. The issue is also where it can make it, how efficiently it can make it. Right. So there are both elements of this problem.
Well, there's higher yields, so costs come down,
Yes.
and there's also speed.
Speed is a big factor too. So E. coli typically batch time is about 24 hours, 18 to 24 hours to ferment a batch. We take 6 to 8 hours. So we are
Right.
able to do 2 to 3 batches in the same time as one batch with E. coli, and our yields are higher. So now, essentially speaking, we can 5x or 10x the productivity as compared to E. coli. That directly lends itself to CapEx and OpEx savings, which reduces the cost of the DNA.
So let's just talk about the practical real world. How plug and play is your system in a real world setting? And I don't know, can you fit into customers' existing infrastructure?
Yeah, that's an awesome question. And that was one thing so I come from capital world. That's what I've done in my life. So from the get-go, one thing we were very clear on, that it should be drop-in compatible. The platform should be drop-in compatible. Otherwise, people have invested hundreds of millions of dollars in not only compatibility in terms of capital equipment, that they should be able to use the existing infrastructure, but also the DNA designs. So it shouldn't require any design changes on the DNA itself, because both
Right.
of those are problematic. So good news is that the technology is plug and play on both ends. We are currently using the E. coli fermenter. We are not developing our own fermenter. It's a commercially available fermenter that people have used for E. coli. Other competitors are using. We are using the same fermenter. It grows really well. On the downstream purification, we are using the same downstream equipment provided by Cytiva or by Sartorius. Nothing changes there. And we actually had a collaboration with Sartorius where we provided our cell paste or our bacteria product to their lab. And we said, can you purify it to Sartorius in their European lab? Can you purify using your equipment that you sell to others for E. coli and tell us if it works? And they did it and they said it works beautifully. So we have results on that. So we have fully confirmed that it's an end-to-end drop-in compatible solution on the manufacturing side of things. Also on the DNA design, we have fully established that it works with all the major backbones and all the major origin of replication and antibiotic resistant markers. And as a matter of fact, we actually are selling our Cyclone, we call it the Vibrio-based DNA cloning strain, where people can clone this DNA in their own labs. And we have shipped to almost 35 plus clients, and the clients are using their commonly used backbone, then they're using E. coli with into Vibrio successfully. And there was a publication out there recently by a nonprofit, I can provide a link later, called Cultivarium, where they actually, without telling us, they bought Vibrio from us and they tested it with various DNA designs, and they found that, yeah, it works. It's a drop-in compatible with E. coli based designs. So we have fully established both drop-in compatibility of DNA designs as well as with infrastructure.
So you said you're getting to cash flow break even next year.
Yes.
So can you talk about that in the context of your customer base? Who are you selling to? I want you to talk about your customer base, but also who you're selling to and how you're going to get wider distribution.
Yeah, so that's where we have spent a lot of our time on. So I come from, actually building profit-making, revenue-generating companies. My other two ventures are all profit-making from day one. And in this business also, that is the intention, that to take it to profitability really quickly. So we have been making revenue for the last 2 years and expanding our customer base. We started making revenue as soon as we had a viable product. And the reason was that's the best way to actually test the market, get feedback from the market, and make money too. So why not, right? So from that perspective, we have 200% revenue growth from 2023 to 2024. We expect to close this year with about 750,000 in revenue, and next year we should break even. Now, on the customer base, we have a variety of customers. We have customers who are CDMOs, who actually make viral vectors, and they buy DNA from us. We also have large, we cannot reveal the details because we have CDA with them, but there are large publicly traded cell and gene therapy companies who use us as their exclusive DNA supplier now, and we have won them from industry leaders in this space because one of the clients put us in a bake-off with the gold standard vendor in this space based out of US. And they gave the same DNA design to us and to them. And we were able to produce that DNA in 3 weeks that they could not in 3 months. And after that, they shifted all their business to us, and we are their prime supplier of DNA. We also are working with hospitals that run clinical trials. So we recently launched two more revenue streams. So we were making R&D grade plasmids for the last 2 years. This year, we launched two more revenue streams. One is a product called Cyclone, which allows users to clone DNA in their own lab. So they can buy the bacteria from us and they can clone it in their own lab. It is a limited use research only license, so they cannot do any commercial activity with it. If they want to do any commercial activity, they have to come to us. So that product was launched in May. We already shipped to 35 plus clients, and the clients include NIH, clients include very large biotech companies and some academic institutions as well. And then the other, go ahead, please.
No. What's the other?
Yeah. So, on the second revenue stream we launched recently is high quality DNA, which is directly used in clinical trials. And there we're working with hospitals that actually make the viral vectors and mRNA, and they inject the client or patients with the drug. So they don't make DNA themselves. They procure it from, service providers like us. So we are working with those hospitals already to kind of provide that high quality DNA, which is a higher quality grade than R&D. So we've added two of these new revenue streams.
Great. Talk to me about the core defensible elements of your IP strategy, and then also, like, how does that interact with your trade secrets? How are you using them both to protect the business?
Yeah, so IP is the kind of the core of our platform. We engaged Greenberg Traurig as our IP lawyer from the very beginning. They're one of the top lawyers out of their Boston office in the biotech space, and they developed our IP strategy. The first thing we did was do a freedom to operate analysis, a detailed freedom to operate FTO, and just to make sure that we are fair and clear of any infringements, which is the case. So our IP strategy is a two-pronged strategy. The first is the Vibrio that we are using is not a wild type. It's heavily engineered. And all the different elements of mutations are put in a patent. And as a matter of fact, our first patent was approved today in the US. So that's a great news for us. And it's a foundational patent. And the good news there is that it blocks anybody else from using Vibrio to make DNA. These are foundational mutations without which the wild type doesn't make DNA very well. So without these mutations, we've blocked the field. And we have filed two more patents on this, which are kind of further iterations of those, I would say the genetic modifications that we have made. So anything that is defensible and enforceable, we are putting it in patents. We can always sequence the bacteria. If somebody goes and does the same thing as we have done, we can sequence the bacteria and enforce that patent. Then the second piece of the IP is trade secrets, where it's, as I mentioned before, it's a white space. Nobody knows how this bacteria works, what kind of media it likes, what is the protocol to mutate the bacteria. Those protocols and those recipes are all trade secrets. And we are way ahead of anybody else. And the reason is my CTO, before joining Novel, had, and very few researchers in the world have that experience, has about 6 years of experience working with Vibrio prior to joining us. So he brought all that knowledge with him. He was the pioneer of Vibrio. He was the guy who published the first paper in Nature on Vibrio.
That's impressive. That's like winning the Academy Award. So it wasn't just a coincidence. Did you search the world and find this guy, or?
Yes. So I read his paper, and that was one of the reasons we selected Vibrio. And so it's an interesting story, and also answers partly why nobody has been able to do it properly. So when we founded Novel, we looked at those papers and we started working with Vibrio. And we had the same problems that anybody else would encounter, that we didn't know what Vibrio likes, how to mutate it, and what mutations to do and how to do them, because there is nothing out there that really, the research papers, there are a few research papers, but when you start doing them in your own lab and you have not done them before, you cannot really do it because you don't have hands-on training. Nobody has done it before. So it kind of prolongs the development. It's a very steep learning curve. So at that point, we got stuck and we didn't know what to do. So I approached Matthew Weinstock, who was the original author, and he had grown in his career and became CTO of Absci, took the company public, and he was coming out of that role, and he was still very excited about the potential of Vibrio, and he joined us, and that fast-tracked our development journey.
Do you have any like specific case studies with customers that illustrate the potential of the platform?
Yes, so we have multiple case studies, and we can kind of provide it. One of them is with the top 5 animal health company. We are pursuing a licensing agreement with them. Term sheet has already been signed. So they basically gave us, their mandate is to make mRNA vaccines for animals. And their major pain points or criteria is that they want to be able to respond fast and the vaccine should be cheap. Those are two main things. Fast, because if there is any outbreak on a farm, they should be able to respond very quickly. And it's for animals on the farm. So obviously cost is a big criteria. So they came to us with 5 different DNA designs, and they said 4 of them they can make, but the yields are low in E. coli. The 5th one is a great vaccine candidate, but we cannot make it in E. coli at all. So we have parked it, we can't take it forward. And so they said, if you can 3 times the yield, make 3 times the yield in your platform, we would like to license it from you. So we put all the 5 designs through its paces with our platform, and we got 10x the yield on every single one of them. And then we went to their lab and did the tech transfer successfully as well. And we were able to make the 5th design as well, and in great quantities, so they could take that design forward. So we just signed a term sheet with them. It's a royalty plus deal, where they want to use our technology globally to make their DNA that will go towards mRNA vaccines. So that's one, I would say, customer case study. The second one I already mentioned, where we were able to make DNA that the gold standard vendor could not make for 3 months. Then we also got another testimonial. I would say we cannot really publish it because it's a government agency. They're not authorized to endorse anybody. But after we launched Cyclone, a cancer researcher was trying to clone a DNA for 6 months using E. coli, and they could not do it because every time E. coli will rearrange the sequence. And with our bug, they were able to do it in 2 days successfully. So we have multiple, I would say, testimonials and case studies coming together.
Yeah, when I was at JP Morgan last year, the one commonality that came out of it was people in the background were definitely nervous about bird flu.
Yeah.
And I got to imagine they're happy you're around.
Yeah, it is, the speed and the cost is a major barrier of the success of these therapies. I mean, we just start, as I mentioned before, we just launched high quality DNA, which goes towards clinical trials. And sometimes, we are all raising money and we are trying to do business, but the bigger picture of this is we are saving lives. And
Yeah.
and the cost is the major barrier to the access of these life-saving therapies. So one example is that after launching our high quality DNA, service, we landed our first PO within 2 weeks, and it was for a compassionate trial for a critically ill or terminally ill patient, and they want to try a therapy on him or her. I cannot reveal the details, but it's a cancer patient, and they were looking for months for a DNA supplier that would meet their budget, and nobody was there, in North America. So when we provided the quote, we were almost one third of the price of the competition because we are very lean and our technology is better, and that allowed them to take the program forward because plasmid DNA is the single largest cost center for these advanced therapies. 30% of the cost of manufacturing is actually for DNA for these therapies. So that really allowed them to move forward, and now that patient has a chance before it. So this is just one example of actually the real impact on people of this kind of technology.
Yeah, that's incredible. That's amazing. So what do you think about your exit strategy? What's your exit strategy?
So the exit strategy is that we have multiple, I would say, exit points, or you can call it inflection points in the business. We are already getting interest from potential acquirers without going into the details where they are interested to look at our technology because we are the only ones with a differentiated bacterial technology in this space, and they are interested to look at us. We are developing a comprehensive data set, and that will be ready in the next 6 to 9 months. And by that time, by the next 12 months, we want to be revenue, neutral. We want to break even. So that will be a very good point for us to look at potential exit scenarios because our technology and the data set will be ready. We will have a longer list of clients with a good revenue number. So we are not looking too far from the exit perspective. It might happen. The exit potential will emerge within the next 12 to 18 months itself. And then it depends from that point on whether we want to exit or not, because with each passing year with revenue increase, the valuation will keep on increasing, right? So we can decide there, or we can delay it for some time to just maximize the return.
Last question, just so I understand. What is the ultimate total addressable market?
Yes,
Is it
the total addressable market right now is about $4.5 billion
great?
between DNA cloning and DNA manufacturing, and it's growing rapidly. So it is expected to be over 15 billion by 2030, 32.
Amazing. Okay, Callie, there's a number of audience questions, so I will let you take it away.
Okay, great. Thank you so much. Are you pursuing any partnerships or collaborations to accelerate commercialization and adoption?
Yeah, so that's a great question. Actually, we are looking at various, there are a couple of aspects there. So for our Cyclone strain, we are looking at partnerships with high volume users. So there are companies that actually make DNA for others in very small quantities, which are not our competition. We make higher quality DNA, high quality GMP, but so the DNA market is divided into various categories. The front end of it is people who make oligos, and then they make very small micrograms of DNA. Those are very high volume producers like Twist and IDT and others. And we're working and talking to them if they can actually get into a partnership with us, because the speed of our platform is a great benefit to them. And we've seen strong interest from them because it can really improve their turnaround time and increase their capacity. So we are working in partnership or developing partnerships with them. The second aspect is on the partnership level is to be the preferred supplier for other CDMOs that make viral vectors or make mRNA therapies, and be the preferred plasmid DNA supplier. And we are talking to various CDMOs and biotech companies on that front. And then the third piece is licensing our technology to end users, not to any CDMOs, like the top 5 animal health company, where they want to make their own DNA and we can license it to them. So we have all these three partnership opportunities that we're pursuing.
That's great. Thank you. And who do you see as your closest competitors, and what prevents them from replicating your performance or taking market share? And I guess part two of that is, talk a little bit about your IP.
Yeah, so the closest or the biggest competitors in this space are the companies that are CDMOs and making DNA. Those are Aldebaran, Charles River, VGXI. Those are US based competitors. In the Europe, Plasmid Factories is one of them. So there are CDMOs that make DNA right now, and they are our competitors. What prevents them from developing a technology like ours is, as I mentioned before, the barriers to entry is really high, and our IP portfolio is getting stronger. We got our first patent already, and that effectively prevents them from using our bacteria that we have engineered to do something else. They are all using E. coli based technology, so they're years away from developing anything that we have. On top of it, we are new and we are very lean on the manufacturing side, because our team is I would like to highlight, is a unique combination of researchers and also people who have expertise in manufacturing. That is very critical in this kind of market where the DNA, different designs of DNA come to us. And if we don't know how to make that, that is a front end work with scientific input. And you need that scientific team input to be able to include that and give it to the manufacturing to manufacture under GMP conditions. So it's both the things working together creates a good customer experience. And our team is very strong in both of those elements. And that's a differentiating factor apart from the technology differentiation that we have.
Great. Thank you. And how are you currently monetizing the technology, product sales, service contracts, licensing, and what does your revenue mix look like in the near term and long term?
Oh yeah, that's a great question. So far, as I mentioned before, most of our revenue is coming from our CDMO operation, where we are making R&D grade plasmids for our clients, and we are using our technology for it. So it is in commercial manufacturing. It is actually being used commercially by us. We launched two more revenue streams this year. One was Cyclone strain, which was launched in May this year, and we've already shipped to 35 plus clients and getting great reviews and repeat orders on that. So that revenue stream is obviously small right now. It was recently launched. And then the third revenue stream we developed is the high quality manufacturing. We got our first PO and actually a very strong response from the market. So today our 90% plus revenue is coming from R&D grade manufacturing services, and the remaining is coming from high quality and Cyclone. Moving forward, we will see that the revenue will next year we see more split between high quality. Cyclone will take bigger share of revenues, and R&D will kind of slow down a bit. So as we move forward, the premium quality manufacturing like high quality and GMP will make the most of our revenue makeup, and other things will be there, but they won't be as prominent.
Thank you. Based on the backlog that you have, how quickly can you scale up operations, and what does that path forward look like?
Yeah, so we already have our scale up path, and that is the beauty of this platform. Because the scales are low, we can actually commercially make high quality and GMP material in a 5-liter bioreactor instead of a 50-liter or a 100-liter required for E. coli. And that essentially means smaller footprint and lower capex and operations, and also relatively very easily to be able to scale up. So we already have a 5-liter bioreactor train running, and that is being used to make high quality material. And the intention with this raise is to go to a bigger lab and hire a bigger team so we can quickly ramp up. Right now, the same team, I have like 8 people, and they're developing the optimization or optimizing the platform. They're also doing R&D grade manufacturing. They're also doing high quality manufacturing, and they're also making Cyclone boxes. So they are being spread very thin, My lab is operating at 120% of their capacity. So the intention of this raise is actually move to a bigger lab and hire a few more people. And then as the market expands, add another line of high quality and potentially a GMP line. And the whole, it is not a very capital intensive maneuver because of the, I would say small scale that this platform operates in.
And then beyond healthcare, are there other applications for this technology?
Yeah, that is actually a great question. So our focus is DNA manufacturing, but we completely are aware that we are one of the few companies, or maybe the only company in the world that knows how to work with Vibrio, and we have scaled it up too. And if you look into the literature, many researchers from Harvard and from even from China, they have really published a lot of studies about how Vibrio can replace E. coli in many different domains because it is a stronger bug naturally. And not only in DNA manufacturing, but in protein expression, waste mitigation, and all those things. So, having said that, May of this year, when we launched Cyclone, was the first time we disclosed to the world that our bug is Vibrio. Before that, we never said. We would say that it's a fastest growing bacteria. We would never disclose it's Vibrio. So in that conference itself, we met uh, the senior vice or very senior executive C-suite executive of a Fortune 200 consumer goods company, and they approached us and they said, "You guys are using Vibrio?" And I said, "Yes." And they were like, "We are trying to use this for the last 2 years to valorize our waste stream because it has a very high salt content, and we want to make something useful out of it because we generate tons of this material every month in every facility we've got." And we have been unsuccessful because of the technical challenges. And those are the same technical challenges that we overcame to launch the product. So once they heard that, they quickly bought a box of Cyclone. They used it, they found it works, and we've entered into a partnership with them to develop that platform technology using Vibrio to valorize the waste stream, which is not our core focus, but they're paying for it, and we are glad to have that partnership. But that opens up another dimension of this technology. Plus, we have a working prototype of protein expression. This bug is great at protein expression, making enzymes and other drugs as well. And I would say expresses similar, uh, I would say better quality than E. coli. We already have that in the working prototype. We'll be launching a protein expression version of Vibrio in the next 6 to 9 months. So that's in the works as well. So yeah, definitely there are other verticals planned. We clearly know that this has bigger potential than just DNA manufacturing.
Great. And then is there a global market for this, and do you have plans for international expansion?
Yes, so there is definitely international market for it. Right now, our pricing is actually lower than even Chinese pricing on the high quality. So if you get a quote from China for high quality, will be similar or slightly lower. So we are talking to clients in Europe, and there are plans to talk to clients in India as well. So and other places. So that's why our IP, we are already entered the PCT stage, and we are registering the IP in other jurisdictions that include China, India, Europe, and Japan as well. So there are definitely plans there, but we want to stabilize the US business first, reach break even before we stretch out.
And, on the heels of that, yeah, so what are your milestones for the next 12 to 24 months? And do you have a preferred exit strategy?
Yes, so the milestones are we need to create, we're in the process of doing it, is there I would say four major milestones. The first one is to create a comprehensive data package with about 10 to 15 different DNAs designs end to end with a full 5 liter scale that shows in comparative to E. coli, that shows the advantages in terms of yield and quality. We already have done 2 or 3 that demonstrates that the technology is scalable, and at R&D we have hundreds of designs that we have done. Like at a shakes flask level, we have done hundreds of designs and compared it with E. coli and demonstrated that we can make that DNA successfully at higher yields faster, and the quality is good. We need we need to create that data set at a 5 liter scale. The process of that has already started. So that's our first objective in the next 9 to 12 months. The second objective, and that includes optimization of the process, the scaled up process. The second objective is to reach break even but in the next 12 to 18 months. And we are on track again, very strong push on business development side. The third, I would say objective we have is to launch our protein expression system. And then the final objective that we have is to explore, beyond our, I would say, North American market avenues and opportunities around it. Now, on the exit strategy, once the data set is ready, we are already getting expression of interest from some of the potential acquirers. We are deliberately waiting. We want to create a full, because we know what they're going to ask for. I want to have that data set ready to go in front of them. And because I have deep network within the industry, before founding Novel, we actually spoke to some of the potential acquirers, the large companies, and we said, if a new technology comes to the market, what would you like to see there? What will differentiate? What will kind of prompt you to do an acquisition? And that kind of has formed the basis of where we want to go. So we should be ready for that within the next 12 months, 9 to 12 months. And my potential exit strategy is I would like to exit, but I would like to exit where I can maximize the returns. So it could be in the next 1 to 2 years, or it could be in 3 to 4 years after we launch formal GMP. That depends on the market conditions and other factors.
Great. Well, thank you very much. And that concludes the questions. For the audience, if you have additional questions or would like to be connected to Novel Bio, please reach out to me. I can connect you. In turn, they will have all of your information as well, and we'll be reaching out. Pankaj, I just want to turn it over to you for any final words before we close out today.
No, thank you so much for the opportunity. We are very excited about this technology. As I mentioned before, it is, more than just business. It is actually saving lives, which is the thing I'm pretty proud of. We already have an example in front of us where we were one of the enablers of a technology or a drug to move forward that could not have been possible without it. And I think we are incredibly capital efficient. That's another differentiating factor for us, apart from the technology, which we're very proud of as well. But we are incredibly capital efficient. So one thing I always say this, that our technology just doesn't exist on PowerPoint. We, on presentations, we are actually using it every day in our lab. Clients are using it, and it has been validated completely. And so from the biotech, and we are revenue making. So from the typical biotech startup perspective, the risk perspective is very different than other investments.
Great. Well, thank you, everyone. And thank you, Pankaj. And I hope everyone has a wonderful day.